In the previous lecture, we explored how primary production introduces energy into marine ecosystems. We saw that photosynthetic organisms, especially phytoplankton, convert sunlight, carbon dioxide, and dissolved nutrients into organic matter that supports nearly all marine food webs. We also examined the environmental conditions that control this productivity, particularly the availability of light and nutrients in the upper ocean. However, phytoplankton populations are not static. Instead, they change continuously through time in response to both biological interactions and physical environmental conditions. In some cases, phytoplankton populations can increase dramatically over a very short period of time, forming what we call plankton blooms. At other times, plankton populations decline as nutrients become depleted or as grazing by zooplankton increases. Today, we examine the dynamics of plankton populations, how phytoplankton and zooplankton interact, how nutrients are cycled within plankton communities, and how physical ocean processes influence these biological systems. Understanding plankton dynamics is essential because changes in plankton populations can ripple through the entire marine ecosystem. One of the defining characteristics of plankton communities is how rapidly they can change. Unlike many larger organisms that require months or years to reproduce, phytoplankton can divide and reproduce on extremely short timescales. Under favorable environmental conditions, some phytoplankton species can double their population in a matter of hours or days. This rapid reproduction means that plankton populations are capable of responding very quickly to changes in environmental conditions. If nutrients suddenly become available in surface waters, phytoplankton populations may increase rapidly. Conversely, if nutrients become depleted or grazing pressure increases, those populations may decline just as quickly. The figure on the left shows chlorophyll levels in orange. Notice how quickly they fluctuate up and down. Chlorophyll fluorescence is a standard metric for measuring phytoplankton populations in aquatic habitats. The two images on the right are taken at roughly the same location, only about 1 month apart. Notice how green the water is during a phytoplankton bloom. The short generation times of plankton are one reason why biological oceanographers often observe dramatic fluctuations in plankton abundance. These fluctuations are not random. Instead, they reflect the interaction between environmental conditions such as nutrient availability, light, and even water temperature. Biological processes such as grazing, competition, and reproduction are also a factor. Because plankton respond so quickly to environmental changes, they often serve as sensitive indicators of ecosystem conditions. Changes in ocean circulation, nutrient supply, or climate variability can quickly influence plankton populations, which in turn affects the entire marine food web. Understanding the plankton dynamics therefore requires examining the balance between phytoplankton growth and zooplankton consumption, as well as the environmental factors that influence both processes. Phytoplankton growth occurs primarily through cell division. When environmental conditions are favorable, a single phytoplankton cell can divide into two daughter cells. Those cells then can divide again, leading to exponential population growth. This pattern of rapid reproduction is one reason phytoplankton populations can expand so dramatically over short periods of time. The rate at which phytoplankton grow depends on several environmental factors, particularly light availability and nutrient concentrations. As we discussed in the previous lecture, phytoplankton requires sunlight for photosynthesis and nutrients such as nitrogen and phosphorus to construct cellular components. If both light and nutrients are available, phytoplankton populations can increase rapidly. However, growth rates vary among different phytoplankton species. Some species are adapted to nutrient-rich environments and grow quickly when nutrients are abundant. Others are better adapted to nutrient-poor conditions and grow more slowly but efficiently when nutrient concentrations are low. Temperature can also influence phytoplankton growth rates by affecting metabolic processes within the cells. Warmer temperatures often increase the rate of enzymatic reactions involved in photosynthesis and cellular metabolism, which can accelerate growth under favorable conditions. So the combination of these environmental factors determine how quickly phytoplankton populations increase within a particular region of the ocean. While phytoplankton are capable of rapid population growth, they are also subject to intense grazing pressure from zooplankton. Zooplankton are small animals that drift in the water column and feed on phytoplankton and other microorganisms. In doing so, they transfer energy captured through photosynthesis to higher levels of the marine food web. Zooplankton grazing plays a crucial role in regulating phytoplankton populations. If phytoplankton populations increase rapidly, zooplankton that feed on them may also increase in abundance. As zooplankton populations grow, their grazing pressure on phytoplankton intensifies, eventually slowing or reducing phytoplankton growth. This interaction between phytoplankton and zooplankton represents a classic predator-prey relationship within the marine ecosystems. The growth of phytoplankton provides food for zooplankton, while zooplankton grazing limits phytoplankton abundance. You can see on the left figure how zooplankton populations trace phytoplankton populations, demonstrating the classic back and forth relationship between predator and prey. These interactions can produce complex population cycles in which phytoplankton populations increase, followed by increases in zooplankton populations, which then reduce phytoplankton abundance. Such interactions are a fundamental component of plankton dynamics and influence how energy moves through marine ecosystems. Under certain conditions, phytoplankton populations can increase dramatically, producing what scientists call plankton blooms. A bloom occurs when phytoplankton growth exceeds the rate at which they are consumed by zooplankton or lost through other processes. Blooms typically develop when environmental conditions become highly favorable for phytoplankton growth. For example, if nutrient-rich water enters the surface ocean through mixing or upwelling, phytoplankton suddenly have access to the nutrients required for rapid reproduction. If grazing pressure is initially low, phytoplankton populations can increase rapidly. Seasonal changes often play an important role in bloom formation. In temperate regions, spring blooms frequently occur when increasing sunlight coincides with nutrients that accumulated in surface waters during winter mixing. These blooms represent some of the most productive periods in marine ecosystems. Plankton blooms are ecologically significant because they represent a sudden increase in primary production. This increase in organic matter can support large populations of zooplankton and higher trophic levels, including fish and marine mammals. It is important to note that not all plankton blooms are good for the food web. Certain plankton can be toxic and cause significant ecological damage when they bloom. One example is Carina brevis, a dinoflagellate that causes red tide events in South Florida. Those of you that visit the marine station will learn more about Carina brevis and other plankton that cause red tide events. However, blooms are often short-lived. As nutrients become depleted or grazing pressure increases, phytoplankton populations typically decline. An important concept in biological oceanography is the distinction between new production and regenerated production. These two terms describe different sources of nutrients that support phytoplankton growth in the surface waters. New production refers to primary production fueled by nutrients that enter the euphotic zone from outside the surface ecosystem. These nutrients typically originate from deeper waters and are transported upward through processes such as vertical mixing or upwelling. Because these nutrients come from outside the surface layer, they represent a new supply of nutrients available for phytoplankton growth. In contrast, regenerated production refers to primary production supported by nutrients that are recycled within the surface waters. As phytoplankton are consumed by zooplankton, or as organisms die and decompose, nutrients are released back into the surrounding water. These recycled nutrients can then be taken up again by phytoplankton. Both processes are important for sustaining marine productivity. In nutrient-poor open ocean regions, regenerated production often dominates because little new nutrient input reaches the surface waters. In contrast, regions experiencing strong upwelling or mixing may support high levels of new production. This distinction is important because new production is more likely to lead to the export of organic matter from the surface ocean to deeper waters, a process that plays a major role in global carbon cycling. Plankton dynamics are strongly influenced by physical ocean processes. Because plankton drift within the water column, their distribution and growth are closely linked to ocean circulation, mixing, and stratification. When water masses mix vertically, nutrients from deeper waters can be transported upward into the euphotic zone where phytoplankton live. This mixing can stimulate phytoplankton growth by replenishing nutrients that were previously depleted in the surface waters. Conversely, strong stratification can limit nutrient supply. When the water column becomes strongly layered due to temperature or salinity differences, mixing between deep and surface waters become restricted. In this situation, nutrients remain trapped at depth, and that limits phytoplankton growth in the surface layer. Large-scale climate variability can also influence plankton populations by altering ocean circulation patterns. Changes in wind patterns, upwelling intensity, or ocean temperatures can modify nutrient supply and therefore influence phytoplankton productivity. One of the most well-known examples of climate-driven changes in plankton dynamics occurs during El Niño events, which we will talk about next. One of the most dramatic examples of climate variability affecting marine ecosystems occurs during events associated with the El Niño Southern Oscillation, often abbreviated as ENSO. ENSO represents a large-scale climate pattern involving interactions between the ocean and atmosphere in the tropical Pacific Ocean. Under normal conditions, the strong trade winds blow from east to west across the equatorial Pacific. These winds push warm surface water toward the western Pacific near Indonesia and Australia. As surface water moves westward, colder, nutrient-rich water rises along the eastern Pacific near the coast of South America through a process known as upwelling. This upwelling delivers nutrients to the surface ocean, supporting highly productive phytoplankton communities along the Peruvian and Chilean coast. These productive waters support large populations of fish, seabirds, and marine mammals. This upwelling and increased phytoplankton production is the reason the eastern Pacific Ocean off South America's coastline is often considered the most productive part of the ocean in the world. This is also the reason so many hammerhead sharks and whale sharks are attracted to the Galapagos Islands. During El Niño events, however, the trade winds weaken or sometimes even reverse direction. Without strong winds pushing surface waters westward, the upwelling system in the eastern Pacific weakens dramatically. As a result, nutrient-rich deep water no longer rises efficiently to the surface. With the nutrient supply reduced, phytoplankton productivity declines sharply. This reduction in plankton abundance can cascade through the entire ecosystem, affecting zooplankton populations, fish stocks, and the predators that depend on them. The ecological consequences of El Niño events extend far beyond changes in phytoplankton populations. Because plankton form the base of the marine food webs, changes in their abundance can propagate through multiple trophic levels. So as we said, during an El Niño event, the reversal of the trade winds shut down the upwelling along the coast of South America. The breakdown of the upwelling prevents important nutrients from being cycled back toward the surface. And as we just said, when that phytoplankton productivity declines due to reduced nutrient supply, zooplankton populations often decline as well because their primary food source becomes less abundant. This reduction in zooplankton availability affects fish species that rely on plankton as a major component of their diet. Many commercially important fish species depend directly or indirectly on plankton productivity. During strong El Niño events, fish populations in some regions can decline dramatically, leading to significant impacts on the fisheries. Predators such as seabirds and marine mammals may also be affected as their prey become less abundant. In some cases, animals may migrate to other regions in search of food, while in other cases, reproductive success may decline due to limited food availability. These cascading effects illustrate how changes in ocean circulation and nutrient supply can ripple through the entire ecosystem. ENSO events therefore provide a powerful example of how physical ocean processes influence biological systems. The dynamic nature of plankton communities plays a central role in shaping marine ecosystems. Because plankton populations respond quickly to environmental conditions, they often reflect changes occurring within the ocean environment. Phytoplankton growth depends on the availability of light and nutrients, while zooplankton grazing regulates phytoplankton abundance and transfers energy to higher trophic levels. These biological interactions occur continuously within the plankton community and influence how organic matter moves through the system. At the same time, physical processes such as mixing, upwelling, and climate variability influence the supply of nutrients to surface waters. Changes in these processes can stimulate or suppress phytoplankton growth, leading to fluctuations in plankton populations. These fluctuations can propagate through the entire marine food web because many marine organisms depend directly or indirectly on plankton. Changes in plankton abundance can influence the distribution and productivity of larger organisms throughout the ecosystem. Understanding plankton dynamics, therefore, provides a critical link between the physical environment of the ocean and the biological structure of those marine ecosystems. Today, we explored how plankton populations change through time in response to both biological interactions and environmental conditions. We examined how phytoplankton growth, zooplankton grazing, nutrient supply, and physical ocean processes interact to shape plankton communities. We also saw how large-scale climate variability, such as El Niño events, can alter nutrient supply and dramatically affect plankton populations. Because plankton form the base of marine food webs, these changes can influence entire ecosystems and even affect global fisheries. In the next lecture, we will expand our perspective from plankton communities to marine food webs. We will examine how energy captured through the primary production moves through different trophic levels and how the structure of marine food webs influence the flow of energy through ocean ecosystems.